Okay, good evening and welcome to the 12th live interaction session. Similar to the previous sessions, what I'll be doing is I'll be going through a set of MCQs and then we'll discuss. I'll give you some time to answer those MCQs and then we'll discuss the solution. So I'll get started now. Uh, so last week, uh, we had finished. We were towards the end of the primary motor cortex, and we had one question that was remaining. So this was the question: which of the following is an effect of a pyramidal tract lesion? A. Ipsilateral paralysis. B. Heightened deep reflex. C. Heightened superficial reflex. D. Flaccid paralysis. E. Hypotonia preceded by hypertonia. F. Claw type clumsy grip. G. Babinski reflex in adults. And H. Contralateral paralysis in most cases. So you can take around 1 minute to answer this, and then we'll have a discussion about this question. You can type your answer in the chat box. Okay, let's see what the response is. Okay, I'll go ahead and discuss the question, MCQ question now. So, uh, first of all, if you recall from last class, we know that the pyramidal tract, a tract is basically a set of neurons that is going from one part of the central nervous system to another. So all the uh, neurons from the cortex that go to the spinal cord and which go to the brainstem together are called the pyramidal tract. So the neurons that go from the cortex to the uh, brainstem is called the corticobulbar tract, and neurons that go from the cortex to the spinal cord is called the corticospinal tract. So they together form a the pyramidal tract. So any lesion or if the pyramidal tract is somehow damaged, then that can lead to a host of disorders or a host of symptoms. And some of these symptoms together is called the pyramidal syndrome. Symptoms uh, resulting from the lesion of the pyramidal tract together is called the pyramidal syndrome. So what are the effects of this pyramidal syndrome? So let us go through them. Ipsilateral paralysis is long. Uh, Ipsilateral is if the lesion is on the right side, then the paralysis is also on the right side of the body. And that is not true. That is what ipsilateral means. So that is wrong. Then heightened deep reflex. We know this. This is true. So the deep reflexes are heightened, whereas the superficial reflexes are uh, suppressed. Then there's heightened superficial reflex. This is false because the superficial reflexes are uh, suppressed. Flaccid paralysis, this is also not correct. Hypotonia preceded by hypertonia, this is wrong. It is actually so hypotonia means hypertonia means an increase in the muscle tone, and hypotonia means a decrease in the muscle tone. So it is the opposite actually. It is first it is hypertonia preceded by hypotonia. That is, first hypotonia happens, then it is followed by hypertonia. So this is also false. Claw like clumsy grip. This is correct. Uh, so the control of fine fractionated movements is lost. So that results in this clumsy grip, claw like clumsy grip. Then there is something called as the Babinski reflex in adults. This is also seen. So if we take a blunt object and we rub it on the foot of a baby, then what the baby does is it extends its toes, and there is fanning of the toes that happens. However, this is not seen in adults. But in the case of pyramidal tract lesion, the same Babinski reflex is seen in adults. So in adults, in normal adults without pyramidal tract lesion, what happens is if we take a blunt object and rub it, has somebody joined? Okay, yeah, I think somebody else has also joined. So if we take a blunt object and uh, rub it on the bottom of the an adult's foot, then there is usually flexion of the toes. But this is not the but in the case of pyramidal tract lesion, this is not seen. Instead, the Babinski reflex is seen. And then heads contralateral paralysis in most cases. This is correct. We have seen that it's not ipsilateral but contralateral paralysis. That means if the lesion is on the right side, then the paralysis is on the left side, and if the lesion is on the left side, then the paralysis is on the right side. So the answer is B, F, G, and H. So now we'll discuss week 8 questions, MCQs related to week 8, which is the cerebellum. Uh, so again, you can go through this question, and then I'll take around 20. I'll give you around 20 to 30 seconds to type your answer in the chat box, and then I'll go through this MCQ. Uh, I'll just set a timer for 30 seconds, and you can type your answer in the chat box. Okay, let's see what the response is. Okay, nobody has responded yet. You can type your answer in the chat box. Uh, the cerebellum is located posterior to the cortex and performs mostly motor functions. It is located posterior to the brainstem and performs mostly executive functions. Uh, Located posterior to the brainstem and performs mostly motor functions. And located behind pons and performs most of the executive functions. So, if you recall that uh, the cerebellum is located posterior to the brainstem, so the option A and option D are eliminated, and it performs. We know that the cerebellum is involved in mostly motor functions. So any problem with the cerebellum will lead to problems in the coordination of the motor system. Uh, so the answer should be C. So you can see this is a diagram of where the cerebellum is. We can see this is the brainstem. So this is the mid. This is the sagittal section of the mid sagittal section of the cerebellum and the brainstem, and you can see that this is the anterior. This particular position is the anterior, and this is the posterior. So it is posterior to the brainstem. Okay. The next question is cerebellar control is contralateral and projects directly to the spinal cord, ipsilateral and projects directly to the spinal cord, contralateral and projects to motor areas that regulate movement, ipsilateral and projects to motor areas that regulate movement. So again, you can take 30 seconds to try and answer this question, and then I'll discuss the MC. Okay, let's see what the response is. Okay, uh, Dr. Nalini has answered D, and yes, so I'll discuss this. Okay, so we know that in the motor cortex, the primary motor cortex, the right side of the hemisphere controls the left movements in the body, whereas the left hemisphere or the left uh, the motor cortex in the left hemisphere controls the movements in the right side of the body because of decussation that happens at the level of the brainstem. So but however, that is not the case with the cerebellum. In cerebellum, the control is ipsilateral, that is on the same side. So the right cerebellum is involved in controlling the right side of the body, whereas the left part of the cerebellum is involved in controlling the left side of the body. Uh, and so the contralateral is removed because that is false. That leaves us with two options: ipsilateral. So it is ipsilateral and projects directly to the spinal cord. This part is wrong because it does not project directly to the spinal cord. It projects indirectly to the spinal cord. And ipsilateral and projects to motor areas that regulate movement. This is correct. So the answer is D. So the response was correct. Uh, so I'll just go through one of one particular slide I wanted to show. Yeah. So we can see that here these are the cerebellar outputs. So uh, So uh, these are the cerebellar outputs. So we can see here that this is the cerebellum and it does not output directly to the spinal cord. So it outputs via the vestibular nuclei and in this case the red nucleus to the interneurons in the spinal cord, but not directly. So that is why that second option was wrong. So these images are from the no, the textbook Candle and Schwartz, Principles of Neural Science. Okay, let us go to the next question. Match the following with regard to cerebellar disorders. So again, you can take 1 minute to answer this, and then we'll discuss this. Okay, let's see what the response is. Okay, Dr. Nalini has responded with C. Okay, let's discuss this. Okay, so we'll 1, 2, 3, and 4. So Astacia is actually uh, just one second. Yeah. So Astacia uh, is actually the inability to maintain a steady limb posture. We have seen this in the cerebellar disorders. Then abasia is inability to produce force to overcome gravity. So 2B. And then dysmetria is overshoot or undershoot during movements. 3D. We have seen this. And this dysdiadochokinesia is inability to perform rhythmic repetitive movements. So 4B. So let us see which one that is. 1, 2A. 1C, 2A, 3D, and 4B. So answer is C. That is correct. Uh, so we have seen that when there is this problem with the motor cortex, then it leads to paralysis and other things, other issues. Depends on the size of the lesion, location of the lesion. But with cerebellum, we can see that whenever there's a disorder or in the cerebellum, it results in uh, loss of coordination of movements to some extent. So that can give us an idea of what the cerebellum is involved in. What is the role of cerebellum in movement? Uh, okay, we'll go to the next one. Which of the following provide inhibitory inputs? Uh, so again, you can take 30 seconds and then we'll discuss this. Okay, let's see what the response is. Okay, okay. So let me go back to one of the slides so that this becomes clear. Uh, so we know this is the situation that is happening in the cerebellum. We have three layers in the cerebellum. One is the molecular layer, the MPG, the other is the Purkinje layer, and the third one is the granular layer. So in the granular layer, we have these granule cells, uh, and there are. It's estimated that more than 50% of the cells in the central nervous system is accounted by the cells in the granule layer. So there are billions of granule cells uh, in this particular layer of the cerebellum. And this receives inputs from various sources, and through these uh, neurons called mossy fibers. So we go here, we can see what the cerebellum looks like. Not able to find that photo. Yeah. So we can see here that there are three distinct layers. So this is the cerebellum. In the cerebral cortex, if you take a small section of it, we can see that there are three layers. One is the uh, granular layer, then the other is a Purkinje layer, and the topmost is the molecular layer. So the granular layer, like I said, receives input, so it's the input layer of the cerebellum. Then in the next layer we have these Purkinje cells. So the cell bodies of the Purkinje cells are in this layer, and then their axons go deep into the cerebellum and synapse with these output structures. Uh, Deep cerebellar nuclei, which are actually output structures. And then the dendrites of the Purkinje cell go into the molecular layer. So thousands of dendrites of the Purkinje cell actually go into the molecular layer, and that is what the molecular layer comprises of. So this is a brief uh, overview of what the different layers are in the cerebellum. So here we can see that we have these various uh, this is what it looks like. So uh, we have these from the inferior olives. We have these uh, cells called climbing fibers, which go and directly synapse with the Purkinje with the dendrites of the Purkinje cells in the molecular layer. So these excite the Purkinje cells, and then the Purkinje cells causes a inhibition in the deep cerebellar nuclei. So this is the inhibition that happens. Similarly, with the mossy fibers, the mossy fibers actually excite the granule cells. The granule cells go in turn excite the Purkinje cells, and then the Purkinje cells cause inhibition or cause inhibition in the deep cerebellar neurons. So Purkinje cells lead to inhibition. That is the key takeaway in the deep cerebellar neurons. And also we have these inhibitory interneurons. So the mossy fiber also synapses with an interneuron called the Golgi cell, and the Golgi cell in turn causes inhibition in the granule cells. So there are various layers of excitation and inhibition that is happening. So let us go back to the question. Which of the following provides inhibitory inputs? So we just saw that the Purkinje cell provides uh, inhibitory inputs to the deep cerebellar nucleus. Then the climbing fiber to the Purkinje system. This is false because the climbing fiber provides excitatory inputs, like we just saw here. It provides excitatory inputs here. So this is false. Then the mossy fibers actually provide excitatory inputs to the granular cells. However, the granules, the Golgi cells, which are the inhibitory interneurons, provide inhibition to the granule cells. So the answer is A and D. Uh, so let me just go back here. So these are Golgi cells, and they provide inhibition. It's not properly shown here, but they get excited by the mossy fibers, and when they get excited by the mossy fibers, they cause inhibition in the granule cells. So there are various levels of excitation, inhibition, and control that is happening. The answer is A and D. Let us go to the next question. Which of the following is a function of Golgi cell? So again, you can take 30 seconds uh, to answer this question. Okay, let's see what the response is. Okay. Okay, so like I just mentioned just now, with regard to the Golgi cell, so this is a Golgi cell, and it's an inhibitory interneuron. So they get excited by the mossy fibers, and then they in turn inhibit. Since they are inhibitory interneurons, they are synapsing with the granule cells, and they inhibit the granule cells. So the answer in this particular question is B, because inhibition to the granule cells. Which of the following are not a function of the mossy fiber? So you can take 30 seconds and try and answer this. Okay, let's discuss. Let's see if there's a response. Okay, no response. Okay, let me just discuss this. Which of the following are not a function of the mossy fiber? So it provides complex spikes of 1 to 3 per second. Uh, this is actually the case of the climbing fibers, where the climbing fibers provide complex spikes of 1 to 3 per second in the Purkinje to the Purkinje cells. So this is not the correct answer. This it is not so which is not a function. So this is not a function of the mossy fiber, right? So this is important. So we'll see what is the function of a mossy fiber first. What is not a function? So this is not a function of the mossy fiber. So this is supposed to be the correct answer, right? This is a function of the climbing fiber. Then they provide excitatory input to deep cerebellar nucleus. Uh, but this is also not correct, right? They don't provide excitation. Yeah, they do provide excitation. So let me just go back to this and show you what happens. So the mossy fiber provides excitation to the granule cells, and they also provide an excitation to the deep cerebellar neurons. And then what happens is the granule cells in turn cause excitation in the Purkinje cells, and then the Purkinje cell causes an inhibition in the deep cerebellar nucleus. So what is happening is there's a comparison of the excitation and the inhibition that is happening at the deep cerebellar nuclei. So the excitation is the inhibition is indirectly caused through the Purkinje cell, through the granule and the Purkinje fibers, and then that is compared with the excitation of the mossy fibers. So that is what is happening. So let me go back to this. So it provides excitatory input to deep cerebellar nucleus. This is actually a function of the mossy fibers. So this is not the correct answer. Then it causes inhibition of the granule cells and Golgi cells. No, this is an excitatory. This is mossy fibers have an excitatory function, so they don't cause an inhibition. So this is the correct answer because it is not a function and brings in information via the inferior olives. It's actually this is not correct. It the climbing fibers bring in information via the inferior olives, not the mossy fibers. So the answer is A, C, and D. Okay, which of the following is false? Again, for this you can take around 1 minute to try and answer this, and then I'll discuss this in detail. Let me set a timer for 1 minute. Okay, let's see if there's a response. So I'll discuss this now. So there are three kinds of inhibitory interneurons in the cerebellum. One is the Golgi cell, the other is the satellite cell, and the third is the basket cell. So the satellite cells basically have the star-shaped cell body, and they have short axons. Uh, so let us see what is the go to the options. Which of the following is false? Basket cells have short axons and receive inhibitory input from parallel fibers and cause excitation to the dendrites of the Purkinje cells. This is false because first of all they don't have short axons, and then uh, they don't receive inhibitory input first of all from the parallel fibers. They receive excitatory input, and they cause here itself it is wrong, and they actually cause an excitation to the cell body of the Purkinje cells. So this is a false statement. Satellite cells have short axons, receive excitatory input from the parallel fibers, correct, and inhibit by synapsing with the cell body of the Purkinje cell. This is false. They synapse with only the dendrites of the Purkinje cells. Uh, then third is basket cells affect Purkinje fibers in such a way that only some of the central fibers cause excitatory effects, and most of them cause inhibitory effects. That is correct. So we know that the granule cell axons actually climb up, go through the in the molecular layer. They become parallel fibers. They travel in the mediolateral direction, and then we have these Purkinje dendrites here in the uh, anterior posterior direction. So they take a 90-degree turn basically, and then we have the satellite cells. So these satellite cells basically get excited by the parallel fibers, and what they do is the overall result, they are situated in such a way that the overall result of this entire system is that only there's only a focused area of the parallel fibers that causes an excitation in the Purkinje cells. So for example, this ends up causing an inhibition, the overall effect, and only a focused area of the parallel fibers cause an excitation. So so that is what the overall effect of the parallel fiber satellite cell Purkinje cell system is. So this is a correct statement. And granule cells excite Purkinje cells, causing inhibition to the deep cerebellar nucleus. This is correct. So which of the following statements is false? A and B is false. So the answer is A and B. Okay. Which of the following is true? Again, you can take 30 seconds to answer this question. I'll just set an alarm for 30 seconds. Okay, let's discuss this. The granule layer is the input layer and receives input from the climbing fibers. This is false because it receives input from the mossy fibers. We have seen this already. Dendrites of the Purkinje cell bodies project to the deep cerebellar nucleus. Wrong. It's the axons of the Purkinje cells that project to the deep cerebellar nucleus. Climbing fiber synapse with the dendrites of the Purkinje cells in the molecular layer. This is correct. That is why it's called the climbing fibers, because they climb through the uh, granular layer and the Purkinje layer and directly synapse with the dendrites of the Purkinje cell in the molecular layer. So in that sense, the molecular layer is called as the processing layer. The granular layer is called as the input layer, and the Purkinje cell is called as the output layer because the axons of the Purkinje cell actually project to the deep cerebellar nuclei, which are output structures in the cerebellum. And then we have the fourth option, mossy fibers provide indirect excitation to the Purkinje cells, whereas the climbing fibers provide direct excitation to the Purkinje cells. That is correct. Because indirectly, because mossy fibers first excite the granule cells, and the granule cells in turn cause excitation in the Purkinje cell. So indirectly, mossy fibers cause excitation in the Purkinje cell, and the climbing fibers, like I already mentioned, they directly synapse with the Purkinje cells and cause excitation. So the answer is C and D. Which of the following is false with regard to Purkinje dendrites? So you can take 30 seconds and then we'll discuss this. Okay, let us see any response. Okay, no response. Okay, let's discuss this. Purkinje dendrites are spread in the medial lateral plane but are very thin in the anterior posterior plane. No, this is false. This is actually the opposite. Purkinje dendrites are spread in the anterior posterior plane but are very thin in the medial lateral plane. This is correct. So let's just open up one of the images. So this is what it looks like. The parallel fibers travel in the medial lateral plane, whereas the Purkinje fibers are spread across the anterior posterior plane. Uh, so there is maximum overlap. There's maximum chance of synapsing of the parallel fibers with the Purkinje cells. So this is what it looks like, the Purkinje cells. In the anterior post direction, it is spread out, whereas if you see it from the medial lateral direction, it's very thin. Okay, this is what it looks like. Climbing fibers provide inhibitory inputs to the Purkinje dendrites. Climbing fibers provide no, this is false. Climbing fibers are excitatory. Climbing fibers converge while synapsing with Purkinje dendrites. That is correct. That is because uh, thousands and thousands of Purkinje parallel fibers synapse with a single Purkinje with a single from the dendrites of the single Purkinje cell. So there's a convergence of so many parallel fibers to a single Purkinje cell. So the answer is B and D. Which of the following statements are false? So you can just take 30 seconds and I'll discuss this question. Okay. see there's a response. Okay, no response yet. Okay, so I'll just discuss this. Neocerebellum helps in which of the following statements is false? Neocerebellum helps in maintaining tone and balance of medial part of the body. This is false. It's neocerebellum, also known as cerebrocerebellum. It's called neocerebellum because it evolved late. Its evolution, it appeared late, uh, uh, and it's seen in humans. Its development is seen in humans, great apes. It's larger in humans when compared to if you compare it to the macaque monkey. And as we go up the evolutionary tree, the size of the cerebrocerebellum or the neocerebellum increases, and it's involved in coordination of movements. So this is not the correct answer. Archicerebellum is the oldest region of the cerebellum. That is correct. So this is also called as a floccular nodular lobe, and it's seen even in the fishes. The archicerebellum is can be seen even in lower animals such as fishes, and it was the first to develop. That was then followed by the spinocerebellum. It's the spinocerebellum emerged after the archicerebellum. Uh, and then finally, the newest is the cerebrocerebellum. Hence, it's called the neocerebellum. And then pontocerebellum is seen in fishes. Pontocerebellum is actually another name for cerebrocerebellum or neocerebellum, and it's not seen in fishes. And neocerebellum is the most recent region of the cerebellum. That is correct. So the answer is B and D. The spinocerebellum outputs via. Uh, so let me just show you that image. So we know that this vermis, this is called the vermis, and this is called the paravermal areas. So this together forms the spinocerebellum. So we can see here that it's outputting through the ventrolateral thalamus, the red nucleus, and the vestibular nucleus. So red nucleus, vestibular nucleus, and fastigial nucleus and interposed nucleus. So let me just open this up. So this is called the fastigial nucleus, and this is called the interposed nucleus. So the answer should be B, C, and D. The dentate nucleus is the output nucleus for uh, the cerebrocerebellum, right? This is the dentate nucleus. Okay, going on to the next question. Which of the following statements are true? Uh, So let me just quickly go through the options. The distal parts of the arm, like fingers, are controlled by large number of interneurons, while the proximal parts, like shoulders, are controlled by the direct cortical monosynaptic projections. This is false. This is actually the opposite. That is true. The distal parts are controlled by direct monosynaptic projections, whereas this is in relation to the motor cortex, and whereas the proximal parts uh, are controlled via the interneurons. Distal parts of the legs like toe are controlled by direct cortical monosynaptic projections, while distal parts of the hand like fingers are directly controlled by large number of interneurons. Again, this is the opposite. The opposite is true. So let us see. Proximal parts of the arm, like shoulders, are controlled by a large number of interneurons, while distal parts like fingers are controlled by direct monosynaptic corrections. That is correct. Both distal and proximal parts of the legs are controlled by large number of interneurons and no direct cortical monosynaptic projections. This is correct. Answer is C and D. So this is one of the most important reasons why we have such dexterity with the hand and fingers. That is because we have direct monosynaptic connections from the motor cortex to the alpha motor neurons that control the hand and finger muscles, and that is why the dexterity of the hand and fingers are the highest, And studying the hand and finger can give us insights into the strategies employed by the central nervous system in control, because the amount of interference is lesser when compared to uh, other control of other parts of the body. Okay. Question 13. Which of the following is false for a cerebellum? It occupies 50% of the volume. This is false. It occupies about 10% of the volume. It contains a special type of cell called granule cell, which contributes to about 10% of neurons in the central nervous system. That is false. The granule cells actually contribute to more than 50% of the neurons in the central nervous system. It's estimated that there are about billions, many billions of granule cells in the cerebellum. It consists of a regular structure. This is correct. And it consists of microstructures that is repeated 1,000 times. This is also correct. The answer is C and D. Okay, which of the following regions of the cerebellum communicate with the vestibular system? A. Flocculonodular lobe. This is correct. B. Paleocerebrum. No, this is not correct. Paleocerebrum is another name for cerebrocerebellum. Uh, Sorry, paleocerebellum is another name for the spinocerebellum, and this is false. The cerebrocerebellum also does not communicate with the vestibular system. And the vestibulocerebrum. So vestibulocerebrum is nothing but the cerebrocerebellum. It's the same thing. It's just another name for. Vestibulocerebrum is nothing but the flocculonodular lobe. It is just another name for it. Uh, so the answer is A and D. Okay, the final question for this week. Select the correct statement. The granule cells provide excitatory inputs to the Purkinje cell. This is correct. We have seen this in one of the images before. Mossy fibers provide excitatory inputs to the granule cells. This is correct. Output from the Purkinje axons is excitatory. This is it is inhibitory. Granule cells provide inhibitory inputs to the Purkinje cells. We just saw this. False. Climbing fibers form excitatory inputs to the Purkinje system. This is correct. Climbing fibers provide inhibitory inputs to the Purkinje system. This is false. Output from the Purkinje axons is inhibitory. This is also correct. Mossy fibers provide inhibitory inputs to the granule cell. This is false. Mossy fibers provide excitatory inputs to the granule cells. So the answer is A, B, E, and G. Okay, so we are at the end of this session now. We have discussed everything. Uh, so thank you for joining in for this particular session, Sanjeev. And if you have any questions, you can ask me now. So this is basically the last session, live interaction session. We are done with 12 live interaction sessions. And we have covered about 8 weeks of content. We have discussed about 8 weeks of MCQ questions up till the cerebellum. So the remaining topics to be discussed are week 9 has half of the cerebellum remaining, and then we have week 10 and 11 have uh, basically forgot what was the next part. Anyways, we have covered 8 weeks of content, and so this should give you an idea of the depth at which we have gone into the topics. So thank you for joining in, and see you. Best of luck if you're taking the exam. Goodbye.